A lot of people have all sorts of questions about iPS cells. You've heard the name before, but you're not quite sure, what actually is this? I'd like to answer that in a way that's easy to understand.
So today, we've put together 15 questions about the things ordinary people are most curious about, including the benefits of actually using iPS cells. Koji, please walk us through each one clearly. Alright, here's the first question. To start with, what even is an iPS cell?
If I had to sum up an iPS cell in one phrase, it's a rejuvenation stem cell. What it really means is that you can get back the cells your body had when it was a fertilized egg. And why does that matter? Our whole body originally grew from that fertilized egg, so it means you get hold of your own cells that can become anything. On top of that, our body grew from just one single cell that multiplied without limit, and iPS cells can multiply without limit in the same way. So you can obtain exactly the type of cell you need, in the amount you need, the cells of your younger self. That's really the remarkable thing about them.
So you can return to your own roots, to your most original state.
Exactly. And what's more, they can become any kind of cell at all.
I see. Thank you. Alright, moving on. Here's the second question.
There are several ways to use iPS cells. The first, as I just mentioned, is that they can become any cell, multiply without limit, and are your own cells. This is what we call regenerative medicine, using iPS cells to make healthy cells that replace the cells in a part of your body that has gone bad, or cells that have been lost altogether. For example, when nerves are damaged by a stroke, or in Parkinson's disease, which was just approved recently, where nerve cells are progressively lost, the idea is that if we can replace them with your own healthy nerve cells, the condition can be treated. That's the kind of thing that becomes possible. Take the heart, for instance. When someone's heart fails, until now some people have had to travel overseas for a transplant. For them, finding a heart that matches their type is extremely difficult. But if we can build heart tissue from their own cells, we can replace it with healthy heart cells. That kind of regenerative medicine is the first use. The second use, because you can obtain any of your own cell types, you can use them to find the drugs that suit you and to learn which drugs are toxic to you. And this isn't only for you. For other patients with the same disease, your cells make it easier to discover drugs that help everyone with that same condition. The third use, which has been getting enormous attention recently, especially in the United States, is longevity, rejuvenation. The reason is that the whole concept of iPS cells is turning cells back to a younger state. Using that concept, people are now working to apply them to rejuvenation and health.
Alright, on to the third question. As for the treatments that use iPS cells as of right now,
This is timely. We're filming this on February 20th, 2026, and just yesterday, for the very first time in the world, Japan approved a product derived from iPS cells.
that's truly wonderful.
It involves making nerve cells from iPS cells to treat a brain disease, Parkinson's, together with a treatment for heart disease, where heart cells are made from iPS cells. Japan being the first in the world to approve these is one example of real-world use. And that's not all. There are many clinical trials underway. By clinical trial, I don't mean just an experiment. We're actually administering these to people and following what happens afterward. This is being done for a wide range of diseases. Age-related macular degeneration, for one, or corneal transplants. Here, we make a clear, transparent cornea from iPS cells and actually transplant it, and through that, eyesight recovers, with safety confirmed as well. Then there's the work at Keio University on spinal cord injury. When you break your spine, you can lose the ability to move. But when these patients receive a transplant of neural stem cells derived from iPS cells, their motor function recovers. In the United States, there's also major investment in cancer treatment using iPS cells. Why does cancer develop? We have cells in our body that attack cancer, but over time they grow weaker, stop attacking it, and that's when cells turn cancerous. When you turn them into iPS cells, as I said earlier, they go back once to a youthful state, and from that rejuvenated state, you create cancer-attacking cells all over again. Because the cells have become younger, their vigor for attacking cancer comes back. This is being researched as rejuvenation immunotherapy, using your own immune cells, made young again, to beat cancer.
When you think about all that potential, it almost sounds like a future where, as long as you have iPS cells, you could do just about anything. Is that really how it is?
Well, I'm not so sure about that. It's a balance with many other technologies. This is just my personal opinion, but to treat the heart, for example, it's probably not only heart cell sheets. At a certain stage of heart disease, a pacemaker may be the better choice. There are standards and options beyond cells. Of course, you can make cartilage cells from iPS cells, so if your knee hurts because the cartilage has worn down, a cartilage transplant is one option. But at the same time, the field of artificial joints keeps advancing too. Depending on the stage and the patient's situation, there's a best choice for each, and iPS cells can be one of the many options you can choose from.
Even just being one of the options, in terms of possibility, the scope is enormous, isn't it?
Yes, absolutely. Having many possibilities is important. For example, take stage 4 cancer, where surgery is difficult. You try the first treatment, and it carries you 4 years. If it comes back after 4 years, the next option buys you another 4. If you have 4 or 5 such options, you can live 20 more years. So someone who's 60 could reach 80, and someone who's 80, which is already close to a natural lifespan, could live to 100. In that sense, having several options on hand is extremely important. Even if iPS cells aren't your first option, they can be a later one, after standard treatment when you need something more, or they can be the very first option. Either way, they expand your treatment choices.
Thank you. All right, on to the next question.
When it comes to the safety of iPS cell-derived treatments, there are several angles to consider. As for the safety of the cells themselves, for these transplanted cells, safety is checked over and over at the manufacturing stage. We establish a process we can trust, test it first in animals, confirm it's safe, and then verify it in small-scale Phase 1 and Phase 2 trials. If those come back safe, then, as happened this time, conditional approval is granted, and it can be offered to more people. Where iPS cells stand today is that we've become able to make stable, safe cells consistently. But still, confirming that each and every batch of the product is made correctly is critically important. And that's true not just for iPS cells, but for any stem cell product. You build an established process that reliably makes them safely. You run that exact same process carefully every time, and at the end, you check thoroughly once more. Safety is secured by those two things together.
I see. Thank you. On to the next question.
There are various ways to make iPS cells. They're genuinely fascinating. You can make them from any kind of cell.
Any cell at all is okay.
That's right. We make ours from blood cells. But the very first time, in Professor Yamanaka's lab, they made iPS cells from skin cells. Of course, you can also make them from the oral mucosa, the lining inside your mouth, from cells inside a tooth, and even from hair. You pluck a few strands of hair, take the cells in the root, and grow them into iPS cells. It's almost like the Monkey King in the old legend, who blows on a strand of his hair and a copy of himself appears. But it really can be done. So, although iPS cells can be made from any cell, the most important question is, which cell can you start from to make them safely and cleanly? That matters a great deal. The reason we choose blood is that you insert a tube into a blood vessel and can draw the blood out under completely sterile conditions. Whereas, if you use the inside of the mouth or hair, you end up with a lot of bacteria present. With recent technology, you can even make them from urine, but you just collect that in a cup, so naturally it picks up plenty of bacteria. So we feel blood is the best choice, and we make our iPS cells from blood. The next step, what do we do with the blood we've drawn? We carry out a process to extract only the cells from it. You spin it in a centrifuge, and the cells separate out by weight. The cellular part separates from the non-cellular part, and you take out just the cell portion. Once you've taken them out, they're still blood cells, so you inject into them the 4 genes that Professor Yamanaka discovered. You introduce these 4 genes into the cells. When you do, at first the cell still thinks of itself as a blood cell. But those 4 genes, as I mentioned, are genes that make the cell believe it's an iPS cell. You put those in, and it takes a few months. The seed of it forms in about 2 or 3 weeks. But becoming a true iPS cell takes several months. Gradually, the cell's sense of itself shifts. It used to think it was a blood cell, but little by little, it begins to forget that. Reaches a, "Wait, what was I again?" state, and eventually recognizes itself as an iPS cell. It really is like that when you watch it. At first, the cell doesn't convince itself it's an iPS cell. The first thing it does is forget. It forgets that it's blood, and only then does it start believing it's an iPS cell. And that plays out over several months. Once that's done, when you have a good number of these cells, the next step is to multiply them. iPS cells multiply endlessly. They double roughly every 16 to 20 hours. So it's double, 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 double. They multiply at an incredible speed. You grow them to a sufficient amount, collect them, and put them into a tiny tube. A tube about this size holds something like 100 million cells. You end up with 100 million of your own cells in one little tube. And you store that in a liquid nitrogen tank at minus 180 degrees Celsius. How cold is minus 180? The home freezer where you keep your ice cream is around minus 10. And since you're opening and closing it, it's really only about minus 4. Even an industrial freezer reaches only about minus 20 to minus 80. Minus 180 is so cold that even an industrial freezer can't produce it. So we use liquid nitrogen. There's that scene in Terminator where they freeze the Terminator. It's that liquid. Nitrogen is basically a gas. So when you turn it into a liquid, it constantly tries to evaporate. And that's how it produces such a low temperature. It's cold enough that anything placed in minus 180 freezes instantly. If you keep the vial of iPS cells we talked about inside it, in theory, you can preserve them indefinitely. Basically, as long as the liquid nitrogen doesn't run out, they don't deteriorate. And here's the interesting part. You take them back out of minus 180 and thaw them in warm water. Once thawed, you start culturing them again, and the iPS cells begin multiplying once more. So, unlike meat, they don't spoil. They're not in a dead state like meat. They're stored alive, and no matter how many years pass, they'll multiply again. That's how iPS cells are made today. It's now been about 20 years since iPS cells were discovered. And there's something that has been pointed out ever since the early days of this research. Personally, I feel we've largely overcome that. In the beginning, the risk of cells turning cancerous was very high, and there were 3 reasons for that. The first, the original method inserted those 4 genes I mentioned directly into the human DNA. And what happens then? DNA contains many vital genes, and when the 4 Yamanaka genes come in from outside and lodge themselves there, they can break the existing genes. So that issue with the 4 externally added genes, the next improvement addressed it. With today's method of making iPS cells, even once they're made, these 4 genes no longer damage the cell's own genes. They now stay on the outside. Being able to make iPS cells that way is the first big advance. The second reason, it's good that the 4 genes no longer insert into the DNA, but if they keep lingering inside the cell, the trouble is that among those 4 genes, there are some related to cancer. Today's technology, though, is remarkable. After you've put in the 4 genes and finished making the iPS cells, you can remove them again. Even with our own iPS cells, if we showed you the finished product, the genes used to make them have vanished so completely that you couldn't tell what they were made with. And the third cancer risk isn't about the quality of the iPS cells themselves. When you make nerve cells from iPS cells, if some iPS cells remain among the nerve cells you've made, nerve cells don't multiply, but iPS cells multiply without limit. This has been addressed by improving the method for turning iPS cells into nerve cells, and by improving the methods that prove no iPS cells remain in the final product. We've now largely reached the point of demonstrating they're not there. That's why clinical trials of iPS cells, trials that put them into the body, have already been approved by both Japan and the United States for more than 10 different diseases, and they're being administered to people. And even so, not a single case has ever turned cancerous. And now we've received approval on that basis. So the risk of cells turning cancerous has, through very, very careful progress, been greatly reduced.
We'll continue in the next video.